Okay, so welcome back to the final session of this first day of our Machine Learning and Science Conference 2020. The final session is a talk by Jakob Macke, who's literally on the verge of moving to Tübingen. He is together with Zeynep Akata. They will be the new professors in the machine learning cluster. And we're very happy to have him also here this afternoon. So by now we should also have our YouTube stream live again. So before Jakob tells you about his work, I will briefly introduce him. So Jakob studied mathematics in Oxford and then obtained a PhD here from the Max Planck Institute for Biological Cybernetics. Before he did a postdoc at the Gatsby unit in London, was a research group leader in Tübingen and Bonn, and then a professor in Darmstadt and Munich. And now since a few weeks, he has come back to Tübingen and joined us here. I think his talk really follows up nicely with Kyle Cranmer's talk from before and shows another sphere of applications for simulation-based inference method in neuroscience. And with that, Jakob, the stage is yours. And all the audience, please remember to ask questions and upvote them, and then I will ask them in the end for you.
Okay, thanks, Philip, for this kind introduction. It's great to be part of this interesting conference. As Philip just mentioned, we're literally in the process of moving to Tübingen, so our moving van is coming tomorrow. So that's why I asked the organizers whether I could have my talk today. I will be talking about some tools we're developing that hopefully help scientists do science better and more easily. In particular, kind of the overall goal of our group is to develop machine learning or AI tools for accelerating scientific discovery. We've had a focus on neuroscience, but we're also interested in other sciences and applications in other sciences. So one of the core kind of ingredients of science is to use mechanistic models. So what do I mean by mechanistic models? Well, mechanistic models, I mean models that are built with the goal of understanding a process. So they're built with some assumptions about the mechanisms underlying the data we're observing. They might have some prior knowledge, for example, from physical principles about the dynamics that govern this process. And they're typically built from parameters that are very easily interpreted. So often these parameters correspond to quantities that can be separately measured. The downside of mechanistic models is that they can be very hard to fit to data because they're designed to give insights. They're not designed to be easily fittable to data. In contrast, machine learning has traditionally machine learning has other focuses and strengths and weaknesses. So the goal of machine learning, at least classically, is not to understand the data, but to maximize some prediction performance, for example. And therefore, these models are not built with assumptions about the underlying mechanisms in mind, but with computation and generalization in mind. And rather than building in detailed knowledge of underlying dynamics, we generally rely on the data to tell us what to do. Obviously, we might have some inductive biases built into the model design. And as a consequence, these models can have many, many parameters, billions of parameters in extreme cases, and often these parameters offer no direct interpretation of what they mean. Obviously, the whole point of machine learning is that these models are specifically designed to be fit to data. So we can see that between these mechanistic models that are classically used by scientists and the kind of models that machine learning people like to use, there's this divide, or these complementary strengths and weaknesses. And part of the motivation for our work is that we want to combine the strength of these approaches to build better tools for data-driven science. In particular, what I will be talking about today is in some ways really a follow-up or special case of the general set of techniques that Kyle Cranmer has been speaking about in his talk. And they revolve around the central question of, given some data or data features, so summary statistics computed from data, how can we find the parameters of a mechanistic model that can reproduce them? So when we're given some data and we want to find a model that can explain that data or can fit that data. At least if you already have a model class in mind. Oh, and I should say that we're particularly interested in stochastic models. So models that have some notion of variability, and this is often implemented kind of through internal random numbers in the model. So you could argue that this question has a very simple answer, and just to do Bayesian inference. If you have a model class and you have some data and you want to find those models from the model class that fit them, then you just have to do Bayesian inference over the parameters. So what is Bayesian inference? Bayesian inference is a general set of techniques that finds those model parameters of a model that are consistent with some data and prior knowledge, the prior. Okay? So for Bayesian inference, you need 3 ingredients. You need a forward model, the model that you use to model your data. You need a prior of parameters that encapsulates the knowledge about what parameters you find plausible a priori, and obviously you need some data. And when you put them into a Bayesian inference approach, then it will give you a posterior, and this posterior will basically tell you which of the many parameters that you thought could explain the data actually fit the data. Okay? So Bayesian inference is, in generality, a very powerful tool for science. However, it does have limitations. In particular, classical approaches to Bayesian inference need the likelihood P of x given theta of the data given the parameters theta. So you need this likelihood function to evaluate, given some parameters and some data, how well, in a sense, these data match this likelihood. And for many mechanistic models that we care about in neuroscience and beyond, we can simulate data easily, but we cannot, or at least cannot easily evaluate the corresponding likelihoods. And Kyle, in his talk, has given a few examples from these kind of very big simulators in physics, but this is also true on a smaller scale for kind of spiking neural network models, for example. It can be very hard to write down a likelihood for it. And moreover, as I mentioned before, we're often interested in doing inference not on actual observed data X, but some summary features X computed from the data. For example, we might only have access to the firing rate of a neuron and not the detailed spike times. And then we want to do inference given the summary feature X, f of X, rather than the full data. And to compute the likelihood, we would have to average over all possible data instantiations that have the same feature, and that can be intractable or at least can be tedious. So we're interested in doing inference in these kind of situations where we assume that we don't have a likelihood. And moreover, we also don't want to assume that the model is differentiable. I mean, many approaches in machine learning heavily rely on things like backpropagation, which require you to propagate gradients through a model. And we don't want to make the assumption that this can be done in our model. There's a general set of techniques, sometimes referred to as approximate Bayesian computation. Kyle has given a big overview over more modern simulation-based inference techniques. I will basically give you one approach that we have been taking that is a strategy that relies on training a neural network, in particular a conditional density estimator, on samples from the model. So in the basic approach of this is that we start off by just generating random parameters from the model. Yeah, so we pick random parameters from the prior, generate random parameters with it. Once we have generated random parameters, we can take our model and generate simulated data sets from that. So this gives us both the parameters that we use to generate the data and some simulated data. Once we have those two, we can use supervised learning to train a neural network on that data. So this neural network takes as input the simulated data and tries to predict the parameters that generated each data point. And this is a particular kind of neural network. It's a neural network that doesn't only output a prediction of what the parameter is, but its output is actually a density. So it, given some data X, it will predict the density of parameters that could have generated it. And this is referred to as a conditional density estimator. And in our case, we will condition this on the input data X, and it will return a distribution over the parameters theta. This density estimator itself will also have some parameters, and we call those parameters phi. So we take some samples from the prior, generate simulated data with it, and then we train a neural network-based conditional density estimator on that to tell us, if I give you some data, tell me what parameters could have generated that. All of this happens on simulated data. Once this is successfully trained on simulated data, we can plug in the empirical data, which we will call X0, into this density estimation network to calculate the posterior. So given the density estimator, it will give us a distribution. And if we do this correctly, this distribution will be the posterior distribution we desire. All right. This process works, but it does have the downside of being potentially inefficient because we might end up wasting a lot of computation time by generating irrelevant simulations. So this can be sped up by closing the loop and adaptively generating new simulations from the posterior once we have an initial estimate of it. And we refer to this general approach as sequential neural posterior estimation, or SNAPE. This general idea was not invented by us. It dates back to so-called regression adjustment approaches in ABC from the year 2002 and 2010. And its modern version, it was kind of invented by George Papamakaris and Ian Murray in a NeurIPS paper from 2016. And we have since developed and improved it in various ways and shown how it can be applied to problems in neuroscience. And we have very recently developed a kind of quite extensive toolbox that hopefully makes it easier for scientists to use these methods. All right. And obviously, if you want to learn about simulation-based inference more generally, about other techniques also, there's an excellent review by Karl Cranmer and colleagues that was published in PNAS this year. Let me skip this. All right, I will try in the remaining time to focus on two example applications of this approach, one in neuroscience, if I have time, one out of neuroscience. So the first application we're going to look at is the so-called stomatogastric ganglion circuit. So this is a famous circuit in the lobster or crustacean stomach, which has been studied extensively in neuroscience. In particular, it has this interesting property that it changes in a nice way with temperature. So when a lobster swims into warmer waters, the stomach doesn't break down, but it just moves slightly faster. Okay, so this is it's been studied extensively in neuroscience to understand what underlying network mechanisms could underrise this temperature, I guess, equivariance. It is also famous for another reason, and it was used as an example to illustrate that in neuroscience you can have very similar data arising from very different parameter configurations. So two very different networks, or networks with very different parameters, could give rise to activity that looks very similar. So we wanted to study this using this inference approach. To do this, we basically seeded SNAPE using a particular data set. And this part is a data trace recorded by Sarah Haddad and Eve Marder. And we feed that into SNAPE. And then we take a big 31-dimensional model in our case, and we try to compute the underlying posterior to it. So this is a posterior distribution over 31 parameters, where the parameters correspond to a biophysical neuron model that's kind of closely aligned with the one by Astrid Prinz that was classically used to study the circuit. There's 3 neuron types in the circuit. So we've got 3 sets of parameters corresponding to the single neuron parameters, and we've got 1 set of parameters corresponding to the synapses between them. So you can do inference on this parameter space. When you will look at it, you will probably kind of ask the question of what you do with a parameter space like that. Well, one thing we can do is just, I'm just zooming in a little bit here, and we can generate data from it. So when we generate simulated data from this posterior, they look very much like the real data. And I should say that we do inference here not on the full data trace, but on some summary statistics, like a bunch of summary statistics, looking at all the different timing between different burst events. This is something that's classically done in neuroscience in this context. So when we sample from this posterior, we get all this realistic data. When we just sample from the marginal distribution, so when we pick parameters where each parameter individually is consistent with the data, but the pairs in the higher order coordinates are not, then we don't get realistic looking data. So this is important that when you design your mechanistic neural network, it's not enough to put every individual parameter into a realistic regime. They all have to be realistic jointly. Okay, so the first question that we went after is the question of if you have 2 very different parameter settings, for example, 2 hypothetical lobsters that have the same neural activity output, but have very underlying network configuration, is there a manifold and parameter space connecting them? So could you interpolate from one of those points to the other one without ever leaving this manifold of parameters that gives you realistic network output? And we can do that by just numerically optimizing for such a path in our posterior. So we start off at the blue dot and ask the question of whether we can get to the orange dot without ever leaving the high probability land. And the answer, at least in this case, is yes. So we can start off, and I'm just zooming in on 3 parameters now, and I'm showing you some examples from this network configuration. So you can start at the orange point and then smoothly interpolate and walk along to the green point, and at every point in between, the network output would look similar. So this tells us that there's directions in parameter space along which are invariant to the network output. We can also ask the opposite question. If you go orthogonally off this manifold or down the gradient, how quickly does the rhythm break down? And the answer is it can break down very quickly. So when we, rather than trying to stay in high probability land, we try to go to low probability land very quickly, then the rhythm very quickly breaks down, right? So we can So this tells us that for this stomachogastric ganglion, there's some reason in parameter space which are possibly designed for the network to be invariant. So we can move a lot around there. We have a lot of redundancy in there without changing the output. But there's other regions where the network is very sensitive and the rhythm completely quickly break down if we perturb it in that direction. Okay. Going back to this full 31-dimensional posterior, when you look at these parameters, and I'm showing a subset of parameters here, you might argue that the posterior looks very broad, right? It kind of looks like the space of possible model is very big. And in this case, this is true. But it gives this illusion that basically any model would work. And also, it also raises the question of whether there's any compensation mechanisms in the posterior. So why is this interesting? Because when people measure these different biophysical quantities from different animals, they often find a lot of correlations. So when one expression has gone up, the other one also goes up. If there's some compensation mechanism, so there's really like a manifold where this lives on, you would expect to get correlated posterior. So why are they so uncorrelated? Well, the thing to remember is that this posterior is really a marginal over 31 different dimensions. Because I can't plot 31 dimensions, I'm showing you the pairwise marginals, which implies that each of those boxes here is an average over 29 dimensions. So instead of looking at this marginal posterior, we basically said, well, what happens if you fix 29 parameters and you just vary the other two? So technically, we're looking at a conditional posterior where we only look at the posterior over 2 parameters and vary everything else. And then you find a lot of correlations in the posterior. And when we do this over different data sets or different data points, in fact, we find that these correlations, in this case is between 2 different channels of the AB neurons, we find that these correlations for different samples are actually quite preserved. So when we sample often, we find that obviously the posterior moves around, but the correlations seem to be quite preserved. There's another example, 2 different samples, similar correlation. Another example, 2 different similar correlations. So what we can then do is to plot all these conditional correlations. And as I said earlier, the marginal posterior looks very uncorrelated, but these conditional posteriors look very correlated, and they look correlated in a way that makes sense. And that all the parameters corresponding to the same neuron or the same neuron type, often they're correlated, but there's very few other correlations. But we can then go in and try to see whether there's experimental evidence that's consistent with these other correlations. And there's and when you look at the few data points that are available, we find that for some of the correlations for which we predict positive correlations in biological experiment, there's some evidence, at least qualitative evidence, that our positive correlations in the posterior correspond to positive correlations in biology. And this is one example here for within neuron parameter, but there's also evidence for synaptic parameters. Okay, I want to switch gears now and move to a second application of this general approach. This is not using straight SNAPE as I described it before, but it's using a similar simulation-based inference approach. This is an application imaging where we looked at a single molecule localization microscopy through the lens of inference. So single molecule localization microscopy is a technique which is basically based on the following idea. You have some biological sample, and this biological sample is very small. So you can't image it. It's basically beyond the classical resolution limit. So rather than imaging it directly, you have some stochastic activation of fluorophores. So rather than everything being fluorescent at the same time, you have these single fluorophores that stochastically activate and inactivate. Then these fluorophores are imaged through an imaging process that involves a point spread function. So you get these blurry images. And then you take these images. From that, you try to extract the underlying fluorophores. And then from the fluorophores, you can reconstruct the biological sample. And that makes it possible to sample biological samples that are kind of smaller than their diffraction limit. So you can get, you can sample nanostructures that you would not be able to do with classical microscopy. And what's clear is that this is inherently a computational technique because there's an important step of, from your image sequence, reconstructing the underlying sources. And we attacked this as a Bayesian inference problem. We just said we have a forward model of the imaging process. So we had a very simple numerical model of the forward process. And then we do Bayesian inference given the images to invert this model and to try to extract the underlying fluorophores. And we, and in particular Arthur Spicer, a graduate student in the group, spend a lot of time optimizing this approach and kind of coming up with special architectures that make this work well. And after he did that, we kind of, this resulted in in an SML reconstruction algorithm that was very powerful or is very powerful. What you see here are two examples taken from a public benchmark data set. And at the top, you see the reconstruction with Arthur's algorithm. Below, you see a reconstruction using a C-spline algorithm, which is kind of one competing algorithm that has been used for that. And I hope you can appreciate by eye that the reconstructions we get with decode, which is what Arthur called his algorithm, are much more crisp than they would be with C-spline in this case. This is also true quantitatively across multiple different data sets. So when we look at this challenge benchmark data sets, there's different data corresponding to high and low SNR data, and there's high density and low density data. What's particularly challenging for these imaging algorithms are high density examples where you might have fluorophores by chance that are close to each other, so you need to, their point spread function will start overlapping. And when you look at all these different data sets, we can see that the black circles, which correspond to different versions of Arthur algorithms, are always substantially show substantially better performance than other algorithms, showing that quantitatively on this public benchmark challenge, it performs well. One should also mention, though, that this public benchmark challenge is on simulated data, and in some ways it has to be on simulated data because that's at least currently the only way to really get ground truth, quantitative ground truth for these for these reconstructions. So we wanted to see whether these advantages also kind of translate to real data. And we teamed up with some kind of real imaging people, in particular Jonas Ries and Lukas Müller from the EMBL in Heidelberg. And they one of the things that they shared with us is a data set where we could generate kind of high density and low density reconstructions. So we could say, like, given a high density reconstruction, can we reference this back to the low density reconstruction, use that as a ground truth? And when we do that, and just zooming in on this particular part here, hopefully you can see that as we increase the density, the performance of decode doesn't drop significantly, whereas the performance of other approaches drops significantly, resulting in these blurry images. Whereas decode still works fine at a higher density, and higher density means you can image more quickly, so you can possibly image dynamic structures. And this is also true quantitatively. You can compute something called the Fourier ring correlation to get an estimate of the spatial resolution. It's not perfect, but it's at least an estimate. And using that measure, we also perform, decode performs better than C-spline in this case. All right. This brings me to the end of the second application. In general, kind of to loop back to what our general goal is, we want to develop machine learning methods for scientists. So one particular thing we have been focusing on is this idea of using simulation-based inference to really make Bayesian inference kind of extended to all kinds of models, including ones for which we don't have likelihoods. The general idea is that we finally get tools that don't require any more hand tuning and that are easy to use for scientists. We're not there yet, so there's still some scientists, some expert knowledge in setting up networks required, but we have very recently released a toolbox which I think in some ways minimizes much of the pain that previous applications of this approach have had. And this was work led by Alvaro Tejero-Cantero and the group. I think it's also important to not only do inference, but to have tools for exploring and visualizing the resulting posteriors. And this idea of searching for these high probability paths was one instantiation of this idea. I will also say that this full black box setting that I kind of phrased this problem in, that we assume we have a simulator which doesn't have gradients, which doesn't have likelihoods, for which you don't have any access to the internals, is very general, but in its generality is always somewhat artificial, because in many cases we have a little bit more or much more information about the simulator. For example, like Kyle mentioned, sometimes you can grab the underlying random numbers, or you can even port your simulator into a framework that allows you to use some techniques from probabilistic programming. So as general as these techniques are, it's important to remember that you can probably often do better by exploiting additional information that you might have. And I also want to be clear that these methods are not a magic bullet or magic wand yet. There's lots of work that can be done, in particular, and needs to be done, particularly making these methods more efficient, more stable, to incorporate better active learning in them, either to find better parameters or better experiments, to make them more robust to misspecification and all sorts of things. So there's a lot of work for us to be done. Let me skip this. Given that this is kind of our first talk in in Tübingen, I also wanted to make sure that I'm highlighting not only our work we do, kind of the few things that I had time to talk about in detail, but there's obviously other research interests that we also have. Some of them involve simulation-based inference. For example, we've used this to constrain large biophysical models. We're using this to analyze connectomics data in collaboration with a group of Marcel Oberländer in Bonn. We're using this to constrain, using this to constrain cognitive models. We're exploring connections between simulation-based inference and GANs. And there's also some problems, in particular ones in neuroscience, where it would be artificial to do simulation-based inference because we have likelihoods. For example, sometimes even for mechanistic models, you can derive likelihoods. And a student, Alexandre René, in my group, together with André Loctin, has been kind of trying to derive approximate likelihoods for some mechanistic models in neuroscience. A second big neuroscience project we have in the lab is to try to understand how objects are represented in the human medial temporal cortex. So we have access to some data from epileptic patients where multiple single neurons were recorded while they were watching Hollywood movies. And now we're trying to use neural network to predict when cells in the medial temporal cortex will be firing in response to these movies and what we can learn about the underlying kind of visual information processing. This is a project in collaboration with Florian Moormann in Bonn, who's providing us the data, and Laura Leal-Teich at TU Munich. And finally, we kind of picked up an old research interest of ours, and that is to look at or develop state space models for neural activity and behavior. And we're doing this in collaboration with in particular, with Aman Salim and Gordon Berman. So with that, I want to thank all of you for your attention, and particularly the many group members that I've been lucky to work with and have been closely involved in this work. Pedro Gonçalves, Jan Mathis Lückmann, and Michael Deisler have been leading and doing much of the work on simulation-based inference in the group. Michael will be coming to Tübingen with us. Arthur Spicer has been leading and running the work on this on the imaging application. David Greenberg has been contributing algorithms on the machine learning side. He now has his own group in Geestach doing machine learning for Earth and climate science. And Alvaro has been very instrumental, in particular, in making these tools from something that works in principle to something that you can download and start using in your research. And luckily, he will also be coming to Tübingen, but rather than staying in our group, he will be heading the Machine Learning Transfer Center. And I'm very excited that this provides opportunities for really trying to make tools that are useful to scientists. So with that, I want to thank all of them and you for your attention. Thank you very much.
Yeah, thank you, Jakob, for the very nice talk and all the different applications that you showed. There's a round of warm applause and a host of questions. So let's get started. So David Clint wants to know, how does this scale? So can you estimate the posteriors for a few thousand parameters? Modest.
Yeah. So no way is the short answer. So at least we're trying to get the full posterior. I'm not saying we get the full posterior perfectly. I showed some examples that some of the higher order stuff we estimate in the posterior doesn't seem to be completely off because the predictive samples are good. But if you want to get the full posterior, it seems impossible to do this in high dimensions, partially because the space, like the full posterior space that could be occupied, grows exponentially in dimensionality. So the applications we focused were typically between 10 and 20, possibly sometimes 30 parameters, and I don't see any path to scaling this to very high dimensional parameters. There might be some tricks you can exploit. So if your parameter space is high dimensional, but the underlying posterior is constrained to a low dimensional manifold, then this might help you in scaling this up. As before, there's some cool recent work by Johann Bremer and Karl Krenmer that has been exploring that direction. I'm not sure how much they've done this for simulation-based inference, but it clearly could be used for simulation-based inference. But in general, we think of these as tools for mechanistic models with a handful of parameters.
Yeah. There's a somewhat related question. So is it possible to obtain coverage properties or frequency coverage properties for the posterior intervals generated by this inference process? So do you know how good the posteriors are?
So we've looked at how good these posteriors are, and the way we've been doing this is in a Bayesian setting, something called simulation-based calibration. where you basically say, I simulate very often from a model with known ground truth, and then you just compare your posterior and look at how often you're right. And if you write too often, then your posterior might be too confident or too broad, but on average, it should give you, the confidence intervals should be exactly as big as they should be. And this is actually an ongoing project by Jan Matthijs Luckman that has been really looking at evaluating and comparing different simulation-based inference techniques. What we find so far is that when your model is well specified, then you find then these techniques, when they have converged, give you they're well calibrated in that sense. This still leaves open the question of, when you apply this to real data, whether your calibration is still trustable there. That's a harder question. But at least in simulated data, as far as we can tell, they're reasonably well calibrated.
Okay, thank you. The next question is by Bedata Goswami. So I hope I understand the question correctly. So given a set of parameters and then two sets of sample data, would it be possible to use the high dimensional posterior distributions to test whether the two data samples are similar or different? So given two data sets, can you use something like the Jensen-Shannon divergence between the posteriors as a measure of difference of the data sets?
So if I interpret this question correctly, it's basically asking, can we use this posterior as a similarity on data? Mm-hmm. And I think it really depends on how much you trust your model, right? If you just want like a similarity measure in data space, then there's no need to fit a model. In fact, it might be counterproductive to fit the model. But if you think this is a good model of the data generation process, and you want to really ask the question of whether your two data sets are generated by the same underlying process, then yes, one can then use the posterior to see whether under this model, whether the same model or similar model could have generated the same data. This is not something we've explicitly done, but something I would see as this is something that should be very possible.
Okay, thank you. Then I think a couple more questions to go. So one by Michael Doran. This is actually a more specific neuroscience question. So in one of the papers of Yves Marder's group on the SCG neurons, they showed that a specific constraint of the parameter is needed to reproduce the data instead of specific values. So can Snape discover these complex relationship between parameters? So something like constraints like parameter A times parameter B must be smaller than some value C?
The short answer is yes. Yeah. And in fact, obviously the work by Yves Marder and Astrid Prinz has been very influential in motivating us to use this somatic gastric ganglion as a way to explore these questions. Is Green still on? Are we still?
Yeah. Your slide is still there.
Okay. Then if I,
we can see it.
So this is just one example here, but if you look at the third last line at the bottom, you see that this is curve. This is this corner curve, and that corresponds to not an individual parameter being constrained, but in this case, this function of the two parameters being constrained. So the short answer is yes. The whole idea is that we can discover these high-dimensional parameter or multi-dimensional parameter constraints, not just constraints on individual parameters.
Okay, maybe stay with those slides around that area, because the next question is also related to that. So it's by Noah Mayem-Shinitsky. So when you estimate these correlations that appear in the conditional distributions, how do you choose the values of the rest of the parameters that you condition on? Okay, so the answer is that we sample from the posterior, and then when we compute correlations, we average the correlations across all the different parameters we picked. And because, as I mentioned before, the correlations look very similar from one parameter set to the other one, it basically doesn't matter whether we average over 100 or 1,000 different parameter sets, these correlations converge quite quickly. So you first compute the correlation, then you average? Yes. Yeah. Okay. Mm-hmm. Okay, thanks. And So I read the question. It's a question by Eric Reidel. So how different are the models that for which one tries to estimate the parameters, or family of models? So they mainly differ in some sets of Sorry, Eric.
So I think I understand the comment. So let me try to
because I haven't yet.
let me ask a question that's hopefully aligned with his question, and then answer the question that I'll ask. So I was kind of cheating a little bit in the sense that I assumed we have a general model class that allows us to kind of decide parameters. But sometimes you might have two model classes. You might say, rather than having one model with many possible different parameter sets, I might have two families of models and they're not jointly parameterized. So how can I decide whether I should use the first family of models or the second family of models? And if we have two families that can be parameterized, we can always construct like a joint model that is a superposition of the two models, where we also learn how much of the first model and how much of the second model we take. So this is a cheap way to do model selection by just combining the different candidate models into a larger family of models, provided this can be done. Eric also is asking what we can do if the family of model is not parameterized, and then I fear we're out of luck. And we always have to work with families of models that we can somehow parameterize in a low-dimensional way.
Okay, I think there was one final question that somehow disappeared, and that was, so which kind of, because you said in the beginning some models that are not differentiable, and I think one person from the audience wondered what kind of models are you thinking of there?
A classical example that is not differentiable are spiking neural networks, right? When you have neurons that emit spikes, and then you can't differentiate through the spiking operation. There's ways to approximately differentiate through spikes, but in principle, you cannot differentiate through the spiking operation. The second way you often get non-differentiability is when you look at your summary statistics. So for example, your summary statistic might be the fraction of neurons that have more than 10 spikes. Yeah. And this sounds a bit artificial, but if you look at kind of neuroscience papers, they often would do selection of neurons, and they would say 10% of neurons had a significant increase in firing rate, which typically means that 10% of neurons had a number of spikes that's larger than a threshold. So if this is the data that you're feeding in, this is the data that's the input to your inference procedure, then it's very hard to differentiate from that fraction you're giving to the underlying spike rates. So this is basically impossible, or at least seems very hard to get anything like a gradient or surrogate gradient through that.
Okay, I think now we answered all the questions. Thanks again, Jakob, for the precise answers and the discussion. And thanks everybody also in the audience for the entire first day of our Machine Learning in Science conference. And thanks in particular to Teresa Authaler and her technical team who really made this possible behind the scenes, who organized the whole day. It has been very, very smooth. So we hope that many of the 142 people that are here with us today will join us again tomorrow. I think we have a really nice program tomorrow afternoon. We will start with Peter Dayan, who became a director at the Max Planck Institute for Biological Cybernetics last year. Dominic Papi is talking about machine learning applications in business. We have another round of spotlight presentations, including some peek into the work of our ethics and philosophy lab. And then Ingo Steinwart from Stuttgart will talk about theory of deep learning. And as a final speaker tomorrow in the late afternoon, Claire Monteloni from Colorado will join us to talk about the role of machine learning for climate informatics. So I think this is going to be a really nice and interesting day tomorrow. So I hope you come back and we'll talk more then. Thanks for joining us, and see you tomorrow.